Hello everyone. Today's topic is plant embryology. This is a very long PPT, so please be patient and stay with us. In this PPT, we will cover these following topics: embryology, parts of a flower, microsporogenesis, megasporogenesis, EMBRYOSAC, pollination, fertilization, endosperm, seed, fruit, parthenocarpy, vegetative reproduction, and micropropagation. So plant embryology is a branch of biology that deals with the formation, structure, growth and development of embryos produced through the union of male and female gametes. Panchanan Maheshwari was an eminent botanist specializing in plant embryology, morphology and anatomy, plant physiology and biochemistry. He was one of the leading plant biologists who established the technique of test tube fertilization of angiosperms. The embryo is the form in which the seedlings of a plant first appears. Understanding plant embryology is important because it provides insight into the genetic and environmental factors that affect the development of a plant. Father of plant embryology in the world, Carl Ernest von Baer, and in India, Panchanan Maheshwari. So reproduction in plants. Reproduction is one of the most important characteristics of all living beings. It is the production of one's own kind. It is necessary for the continuation of the species on earth and also to replace the dead members of the species. The process by which living organisms produce their offsprings for the continuity of the species is called reproduction. The modes of reproduction vary according to individual species and available conditions. It may be simply by division of the parent cell, as in unicellular organisms, by fragmentation of the parent body, by formation of buds and spores, or it may be very elaborate, involving development of male and female reproductive organs. Irrespective of the mode of reproduction, all organisms pass on their hereditary material to their offspring during the process of reproduction. In this lesson, you will study about the process of reproduction in plants. After completing this lesson, you will be able to define reproduction, differentiate between vegetative, asexual and sexual reproduction, describe the methods of asexual and sexual reproduction in lower plants and filamentous green algae, describe the mode of reproduction in flowering plants, explain the parts of a dicot flower and their functions, describe stages of microsporogenesis, depict the help of diagram the structure of ovule and mention the steps of megasporogenesis, describe the stages of development of gametes in flowering plants, state the types of pollination, their significance and various modes of pollination, explain the steps involved in fertilization, embryo development, endosperm development, formation of seed, differentiate between structure of dicot and monocot seeds, explain the formation of fruit and parthenocarpy, describe seed germination, define vegetative reproduction, differentiate between natural and artificial propagation, explain the advantages and disadvantages of vegetative propagation, describe the role of tissue culture technique in micropropagation and state the advantages of micropropagation. So the various modes by which plants reproduce are of three types: asexual, vegetative and sexual. In asexual mode of reproduction, offsprings are produced from a vegetative unit produced by a parent without any fusion of gametes or sex cells. A single parent is involved. Offsprings are genetically identical to the parent. Asexual reproduction may be of the following types: fission, as in unicellular organisms like bacteria and yeast, where the content of the parent cell divides into 2, 4 or 8 daughter cells, and accordingly the fission is known as binary or multiple fission. Each newly formed daughter cell grows into a new organism. Budding. A bud-like outgrowth is formed on one side of the parent cell, and soon it separates and grows into a new individual, like in yeast. Fragmentation in filamentous algae and accidental breaking of the filament into many fragments. Each fragment may give rise to a new filament of the algae by cell division. Spore formation in lower plants, including bryophytes and pteridophytes, special reproductive units develop asexually on the parent body. These are called spores. They are microscopic and covered by protective wall. When they reach the suitable environment, they develop into a new plant body. Example: bread mold, moss, fern, etc. Vegetative reproduction, it involves formation of new plantlets from vegetative cell, buds or organs of the plant. Here a vegetative part of the plant gets detached from the parent body and grows into an independent daughter plant. It is similar to a sexual reproduction in that it also requires only mitotic division. No gametic fusion, and daughter plants are genetic clones of the parent plant. We will discuss the different type of vegetative reproduction in endosperms later in this lesson. Sexual reproduction involves fusion of male and female gametes, which are haploid, produced by male and female reproductive organs. This fusion is known as fertilization and results in the production of zygote. Further development of zygote gives rise to a new individual, which is diploid. Here at some stages of the life history, meiosis is involved and the offsprings are not genetic clones of their parents. Reproduction in lower plants. We will study the different types of reproduction in two lower plants, one unicellular algae Chlamydomonas and the other is multicellular filamentous algae Spirogyra. Only for your knowledge, Chlamydomonas, it is a haploid unicellular algae found in fresh water ponds. The plant body is pear-shaped with two flagella attached at the narrow end. On one side of the cell, an eye spot is present. A large cup-shaped chloroplast is present. Towards the center, a definite nucleus is present. Chloroplast contains a single pyrenoid. Reproduction. Asexual reproduction with the help of zoospores. Chlamydomonas loses flagella and becomes non-motile. Its protoplasm divides mitotically and forms 4 to 8 zoospores. The parent cell wall is ruptured and zoospores are released. Each zoospore develops a cell wall and grow into an adult cell. The parent cell does not exist anymore. Sexual reproduction. The cell becomes non-motile by losing its flagella. The protoplasm divides mitotically into 2, 4, 8, 16, 32 daughter cells. Each daughter cell develops flagella and is released in water by the rupture of mother cell wall. It acts as a gamete. The gamete is morphologically identical in structure, but all identical, it means same. Gametes released from two different mother cells fused together. The contents of the gamete fuse and form a zygote. This is the only diploid stage in the life cycle of Chlamydomonas. The zygote develops a thick wall around itself to tide over unfavorable conditions. On the return of the favorable conditions, the diploid nucleus divides by meiosis and forms 4 haploid zoospores. Each zoospore grows into a new adult Chlamydomonas. You can see this in the figure and compare them. Next is Spirogyna, a multicellular algae. It is a free-floating algae found in freshwater ponds. The body has a row of rectangular cells joined end to end. Each cell has a spiral ribbon-shaped chloroplast with many parinoids. Central region has a large vacuole. The nucleus is present in the center of vacuole, supported by cytoplasm. Reproduction. Vegetative reproduction by fragmentation. The filament breaks into small fragments and each fragment grows into a new filament by cell division. Scalariform conjugation. Two filaments come to lie very close to each other. Cells of the two filaments form a contact with the help of a tube called the conjugation tube. Cytoplasmic contents of each cell round off to act as a gamete. Gamete from one cell passes to the other cell through the conjugation tube. Each filament acts either as male or female. The contents of two gametes fuse in the female cell and form a diploid zygote. The zygote develops a thick wall around itself and tides over the unfavorable period. On the return of favorable conditions, the diploid nucleus divides by meiosis into 4 haploid nuclei. 3 of these nuclei degenerate, only one remains functional. On germination, wall of the zygote ruptures and a small tube-like structure containing one haploid nucleus comes out. The small tube develops into one filament by repeated mitotic divisions. Now, reproduction in angiosperms. Angiosperms reproduce both by vegetative as well as by sexual methods. In this section, we will study the sexual reproduction in angiosperms. As you know, sexual reproduction occurs by fusion of male and female gametes present in the flower. Thus, flower represents the reproductive unit of a plant. How frequently do plants flowering? There is variation shown by the plant in this respect. Angiosperms can be classified as annuals, biennials and perennials, depending upon the time they take to complete the life cycle and flowering. Annuals, plants live for 1 year. The plants which produce flowers and seeds with one season are called annuals. Biennials, plants which complete their life cycle in 2 seasons are called biennials. In the first year, the plant remain in the vegetative state. In the second year, they produce flowers, fruits and seeds and then die. Perennials, plants which live for several years are termed perennials. Their vegetative stage may last from 1 to a few years, after which they produce flower, fruits and seeds every year, mango, peepal, neem, etc. Monocarpic, however, some perennial plants reproduce only once in their lifetime and then die. Such plants are called monocarpic plants. Initiation of flowering. As the seed germinates, a new plantlet emerges from it. The young plant grows vigorously and continues to grow till it attains a definite shape and size with its vegetative part well developed. This phase of the life cycle represents the young or the juvenile phase. Then at a certain point of time, on completion of vegetative growth, the plant switches over to its reproductive phase or adult phase and vegetative shoot apex transforms into a reproductive or floral apex and starts bearing flowers. As listed above, this transition from vegetative, the flowering stage may take several years in trees, but only a weeks or days in annuals. Juvenile and adult, you can see the difference. Juvenile or young shoot is very small, soft stem bearing a few young leaves. Shape and size of leaf remains same. Shoot does not respond to stimuli to produce flowers. An adult shoot, well developed stem and leaves. Shape and size of leaves change. Shoot respond to stimuli to produce flowers. In cereals, a minimum of 7 leaves must be developed before the plant can produce flowers. Factors affecting flowering. Flowering in a plant is affected by temperature and light. Vernalization, low temperature treatment which stimulate early flower formation is called vernalization. Photoperiodism, it is the response in growth and flowering of a plant to the duration of light and dark period per day. Sex in flowers, you have studied in lesson 5 on shoot system that flowers may be bisexual or unisexual. In some dioecious species, there may be a chromosomal basis of sex determination. For example, XX and XY chromosomes. The male and female plants may also exhibit differences in the level of their growth substances. For example, plants of cucumis which bear male flowers have a high gibberellin content as compared to those which bear only female flowers. The application of gibberellin from outside can induce the formation of male flowers even in genetically female plants, and treating male plants with auxin or ethylene may develop functional female flowers. The above response has also seen in cannabis. Parts of a flower. As you have already studied, a typical flower consists of 4 holes borne on a thalamus or stalk. These holes are from outside. Calyx consisting of sepals. Corolla consisting of petals. Androecium consisting of stamens. Gynoecium consisting of carpels. So try to recollect their role in reproduction. The 2 outermost holes are known as non-essential or accessory holes, as they aid in reproduction but do not directly take part in this process. The other 2 holes, androecium and gynoecium, are known as essential holes, as their absence from flower will lead to failure of reproduction. This is the structure of flower. You can see here calyx, corolla, androecium and gynoecium. So stamen, microsporangia and pollen grain. Stamen consists of an anther containing 4 pollen sacs or microsporangia supported by a cylinder filament. Sorry. Each sporangium contains mass of large cells showing prominent nucleus and abundant cytoplasm. These are the sporogenous cell or the microspore mother cell. Each microsporangium, when mature, has a wall made up of distinct layer of cells. Outermost layer is epidermis, middle layer of thin walled cells, innermost layer the tapetum consisting of large cells which nourish the developing pollen grains. Development of male gametophyte. The wall of the microspore consists of 2 principal layers. Outer axine with some thin spaces. Axine is made up of extremely durable substance called sporoprollenin. The pollen tube grows out of the pollen grain through the germ pores. Inner thin cellulosic wall, this is called intine. The microscopic nucleus move toward periphery and the cell divides into a large vegetative cell and a small generative cell. The pistil, megasporangium and embryosac. The main part of the ovule is enclosed by two integuments, leaving an aperture. This open area is called micropyle. The ovule is attached to ovary by a stalk. Chalaza is the basal part. Female gamete. The gynoecium or pistil represents the female reproductive part in the flower. Each pistil consists of stigma, style and ovary. The ovary contain one or more ovules, which are the future seeds. An ovule develops as a projection on the placenta in the ovary. It consists of a parenchymatous tissue called the nucleus and is covered by one or two covering called integuments. The integument surrounds the nucellus all around but leave a narrow passage, the micropyle, through which a pollen tube may enter at a later stage. As the ovule grows, it is raised on a stalk-like structure called funiculus, which is attached to the placenta on the other end. Development of female gametophyte. Within the nucellus, a single hypodermal cell enlarges and becomes the megaspore mother cell. The mother cell undergoes meiotic divisions and gives rise to 4 haploid megaspore. Usually 3 of them degenerate or disintegrate and the remaining one becomes the functional megaspore. It enlarges and its nucleus undergoes 3 successive mitotic divisions. As a result, 8 haploid nuclei are formed. This enlarged oval-shaped structure with 8 haploid nuclei is referred as embryosac. These nuclei then migrate and get arranged into 3 groups. 3 nuclei reach the micropylar end of the embryosac and other 3 move in the opposite direction and the remaining 2 is the center. The cell membrane and cell walls develop around the nuclei excepting the 2 at the center of the embryosac, which now is called the central cell. The nucellus contains embryosac. Thus, in a mature ovule, the embryosac contains 8 haploid nuclei, but only 7 cells, 3 cells at the micropylar end from the egg apparatus and the 3 cells at the chalaza end are the antipodal cells. The remaining 2 nuclei called the pollen nuclei may use to form the diploid secondary nucleus. In the egg apparatus, one is the egg cell and remaining 2 cells are the synergids. A fully developed embryosac with the nucellus, integuments and funiculus together constitute the mature ovule. In this condition, the ovule awaits fertilization, which must be preceded by pollination. Function of cell and nuclei of embryosac. Secondary nucleus during fertilization. The secondary nucleus fuses with one sperm to form a triple fusion nucleus. This is called primary endosperm nucleus. It gives rise to the food storing endosperm of the seed in many plants. Egg cell fuses with the second male gamete to give rise to the zygote, which develops into the embryo. This is called double fertilization. Synergid cells considered to help in fertilization by directing the pollen tube to the egg cell. Antipodal cells, there is no function. The embryo sac, also known as the female gametophyte, is an oval structure found in flowering plant ovules. When the haploid megaspore nucleus splits, an embryo sac is said to develop. It has 2 haploid nuclei and 6 haploid cells that are devoid of cell walls. The haploid nuclei can sometimes merge to create a single cell. One male nuclei and one egg nuclei join during fertilization to produce a zygote, which leads to the development of the embryo, the primary endosperm nucleus and the second male nuclei fuse to produce the endosperm. So, what is embryo sac? Megaspore mother cell, also known as megaspore, are diploid cells in plants that undergo meiosis and produce 4 haploid megaspores. A haploid female gametophyte develops from at least one of the spore. The megaspore mother cell develops from the megasporangium tissue. The megasporangium is also known as the nucellus in flowering plants, while the female gametophyte is also known as the embryo sac. The female gametophyte is angiosperms is a small structure with only a few nuclei. The cytoplasm associated with these nuclei is not partitioned by cell walls. In sexual reproduction, one of the many nuclei of the embryo sac acts as the egg, combining with one of the 2 sperm nuclei provided by the pollen tube. The second sperm nucleus from the pollen tube fuses with 2 additional nuclei of the embryo sac. This triple fusion nucleus is the predecessor of the endosperm, which is a multicellular food storage layer of the seed. Double fertilization is the process of 2 nuclei from the pollen tube fusing together. This is the most distinguishing character of angiosperm, and it is not seen in any other group. Gymnosperms, on the other hand, have a multicellular female gametophyte with hundreds, if not thousands of cells. In this situation, there is no double fertilization and the female gametophyte develops into the seed's food storage tissue. It is made up of 2 haploid nuclei and 6 haploid cells without cell wall, 2 synergids, 3 antipodal cells and 1 egg cell. Polar nuclei. The 2 haploid polar nuclei can sometimes fuse together to generate a single endosperm mother cell. One male nucleus combines with the egg nucleus during fertilization to produce a zygote, which develops into the embryo. The endosperm nucleus is formed when the second male nucleus unites with the primary endosperm nucleus. The endosperm is formed when this is split. So, embryo sac formation, megagametogenesis, is the process of the female gametophyte maturing in plants, during which the megaspore emerges from megasporogenesis and develops into the embryo sac, which houses the female gamete. During the process of megasporogenesis, the growing embryo goes through meiosis, which involves many phases. The surviving megaspore continues through mitosis, resulting in a structure with 2 nuclei known as binucleate embryo sac, with each nucleus migrating to opposing ends of the embryo sac. 2 rounds of mitosis occur in the haploid nucleus, resulting in 4 haploid nuclei on either end of the embryo sac. Each set of nuclei in the embryo sac migrate to the center to create the binucleate endosperm mother cell, leaving 3 nuclei on the micropylar end. The pollen nuclei, egg cell and synergids are surrounded by cell plates. So, the megagametophyte is produced by 2 separate processes from the megaspore mother cell: megasporogenesis and megagametogenesis. Megasporogenesis is the process of producing megaspores from a megasporocyte that has gone through meiosis. Because of the 4 megaspores that are formed and add value to the female gametophyte, the meiosis of megasporophyte nucleus results in the development of haploid megaspore nuclei, and the sequence is known as monosporic megasporogenesis. After the initial meiotic division, cytokinesis occurs, resulting in cells with 2 haploid nuclei in each of them. Megagametogenesis. The embryo sac is created when a functioning haploid megaspore undergoes mitosis to produce an 8-nucleate, 7-celled gametophyte. Out of all 8 nuclei, the polar nuclei travel to the center to join and form a single diploid cell at this site. When this single diploid cell joins the sperm, the triploid endosperm is created. The other 3 nuclei develop into antipodal cells, while 2 develop into synergid cells that progressively degenerate. So this is the structure you can see in the diagram: 1, 2, 2 to 4, 4 to 8, 3 on the one side, 3 on the other side, and 2 reach on the center. So classification of embryo sac. Based on the number of megaspore, embryo sac can be divided into 3 types: monosporic, bisporic, tetrasporic. Monosporic, meiosis of diploid megaspore mother cell in the nucellus generates 4 haploid megaspores in the monosporic or polygonum type embryo sac. 3 of the megaspores, generally those at the nucellus micropylar end, die as a result of programmed cell death, leaving only one functioning megaspore. Bisporic, due to the absence of cytokinesis and cell plate formation, after the second meiotic division in bisporic embryo sac, meiosis creates only 2 megaspores, each containing 2 haploid nuclei. The megaspore closest to the micropyle then dies, leaving just a single functioning megaspore with 2 haploid nuclei. Then tetrasporic embryo sac have a single 4-nucleate megaspore because cell plate fails to develop during both meiotic divisions. So because bisporic and tetrasporic embryo sacs are formed from 2 or 4 separate meiotic products, their nuclei are not genetically similar to those of monosporic embryo sacs. Then anatomy of the mature embryo sac. Egg cell. The egg cell is found near the embryo sac micropylar end and eventually combines with the sperm nucleus to form a zygote. The egg cell is in close proximity to the 2 synergids, separated from them only by the plasma lemma or partial cell walls. Due to the existence of a huge vacuole at the micropylar end that confines the nucleus and most of the cytoplasm to the chalaza end, the distribution of cytoplasm within the egg cell is highly polarized. Next is synergids. The synergids, which are found on either side of the egg cell, are crucial in fertilization. Before the sperm nuclei are incorporated into the egg and central cell, the pollen tube releases its contents into one of the synergids, center cell. This cell, which is found in the heart of embryo sac, has 2 nuclei, a huge vacuole, and numerous cytoplasmic organelles. The polar nuclei of the serocytic megagametophyte begin at both the micropylar and chalaza ends and move to the center during cellularization. Next is antipodal cells. At the chalaza end of the embryo sac, 3 antipodal cells are situated opposing the egg. The antipodals have not been assigned a defined role during reproduction. However, they are considered to be involved in the import of nutrients to the embryo sac. Embryo sac use. It is the megaspore of a seed-bearing plant that forms the egg cell or nucleus from which the embryo plant grows following fertilization and gives rise to the endosperm. Conclusion: an integrated approach to studying the female gametophyte of angiosperms reveals fresh information on the mechanics of sexual reproduction in higher plants. To answer concerns generated by morphological investigations, researchers are using genetic, molecular, and physiological techniques. These approaches have the potential to uncover new knowledge on ovule and embryo sac development ovule-embryo sac interactions and the biology of the female gametophyte. So you can see in the diagram monosporic polygonum type, bisporic elisma type and tetrasporic drusa type. This is the chart. Female gametophyte is divided into three categories: monosporic, bisporic and tetrasporic. Monosporic has two subtypes, bi also two subtypes and tetrasporic has 7 subtypes. In monosporic polygonum oinothera, in bisporic allium, endymion and tetra में 7 subtypes, but fritillaria is important. This is the complete structure. You can see open area is called micropyle, closed is called chalaza and integument, and you can see 8 cells and 8 nucleus and 7 cells. So they defined as an integument megasporangium. It encloses the embryo sac, which is the female gametophyte of angiosperm. Types of ovules. Ovules are of 6 types. First one is atropous or orthotropous. The ovule is straight. You can see this in the next slide. Diagram of this orthotropous ovule. The ovule is straight so that the micropyle lies on the same vertical axis with the funicle and chalaza. Polygonum, piper. Anatropous in this type, the body of the ovule becomes completely inverted so that the micropyle and hilum come to lie very close to each other. The micropyle and chalaza lie on the same vertical axis but not the funicle, castor and helianthus. Campylotropous, when the ovule is curved so that the micropyle and chalaza do not lie on the same straight line, it is called campylotropous. Amphitropous, when the ovule's curvature is so pronounced that the embryo sac bends like a horseshoe, the ovule is called amphitropous. Hemianatropous, in this type, the nucellus and integuments lie more or less at right angles to the funicle. Cercinotropous is last one in this type. The nucellus protuberance is at first in the same line as the axis, but the rapid growth on one side makes it anatropous. The curvature continues until the ovules turn entirely, with the micropylar end again pointing upward. So here is the diagram: atropous or orthotropous, anatropous, amphitropous, hemianatropous, campylotropous, cercinotropous. Then pollination. When mature pollen grains released from anther are carried to stigma of a flower of the same or different species, it is called pollination. Pollination, transfer of pollen grains from the anther to the stigma of flower. Pollination is of two types: self-pollination, transfer of pollen grains to stigma of same or another flower of same plant as in the China rose. Cross-pollination, transfer of pollen grains to stigma of a flower of another plant of the same species and as in palm. Importance of pollination: it results in fertilization and ovule is converted into seed. New varieties of plants are formed through new combination of gene in case of cross-pollination. During pollination, pollen tube produces growth hormones which convert ovary into fruit. Cross-pollination is brought about by various external agencies such as wind, insects, water, birds and other animals. Now let us study the various agencies of cross-pollination which carry pollen grains from one flower to stigma of another flower. So pollination by insects. This is called entomophily. Flowers are usually large, colored and showy to attract insects. Some of these flowers secrete nectar to attract insects. Salvia flowers show special adaptation for pollination by bees. You can see in the diagram. Characteristic in flowers which favor cross-pollination. Pollination by wind. This is called anemophily. Flowers are small, without color, nectar and scent. Flowers produce a large number of pollen grains to allow for wastage when carried with wind to another flower. The pollen grains are small, light and sometimes provide with wings. The stigmas are comparatively large, protruding and sometimes hairy to trap pollen grains, for example, grasses and some cacti. Pollination by water, this is called hydrophily. This takes place in aquatic plants. Pollen grains are produced in large numbers. Pollen grains float on surface of water till they land on the stigma of female flowers like Hydrilla, Vallisneria. Pollination by animals, zoophily. Flowers of such plants attract animals by their bright color, size, scent, etc. For example, sunbird pollinates flowers of canna, gladioli, etc. Squirrels pollinate flower of silk cotton tree. Humans carry out artificial pollination in a number of plants for producing hybrids. Some adaptations to promote cross-pollination. Unisexuality, flowers may be only male or female, born on different plants like papaya, palm. Dichogamy, male and female sex organs mature at different times in sweet pea and salvia. Anther matures earlier than the stigma, and in custard apple, carpel mature earlier than the anther. Self-sterility, pollen grains are incapable of affecting fertilization even after being placed on the stigma of the same flower. Devices to ensure self-pollination, cleistogamy, flowers do not open completely on maturity. Male and female sex organs mature at the same time. Next is fertilization. Everyone knows about fertilization, fusion between male and female gametes. Very simple definition. Pollen grains reach the right stigma and begin to germinate. Pollen grains form a small tube-like structure called pollen tube, which emerges through the germ pore. The contents of the pollen grain move into the tube. Pollen tube grows through the tissues of the stigma and style, and finally the ovule through the micropyle. Vegetative cell degenerates and the generative cell divides to form two sperm or male gametes. Tip of pollen tube bursts and the two sperms enter the embryo sac. One sperm fuses with the egg and forms a diploid zygote. The other sperm fuses with the secondary nucleus to form the primary endosperm nucleus, which is triploid in nature. Since two types of fusion, syngamy and triple fusion, takes place in an embryo sac, this process is called double fertilization. After triple fusion, primary endosperm nucleus PEN develops into an endosperm. Endosperm provide food to the developing embryo. Later, the synergids and antipodal cells degenerate. Significance of fertilization: it gives stimulus for the growth of ovary, leading to fruit formation, help in recombination of characters as genes from two different individuals combine and form the zygote. Post-fertilization change: events that follow double fertilization are development of endosperm and embryo and maturation of the ovule into seed and ovary into fruit. Endosperm, this is of three types. The endosperm development begins before embryo development. Can you tell why this is to establish the nutritive tissue for the growth of the zygote into an embryo? The primary endosperm cell divides repeatedly and forms an endosperm tissue. There are three ways in which the endosperm may develop. Number one, nuclear type. The nucleus undergoes repeated division to give rise to free nuclei, which arrange themselves at the periphery, leaving a large central space. Cell wall formation starts subsequently and endosperm becomes cellular at maturity. This is the most common type of endosperm development and is seen in maize, wheat, rice, etc. In cellular, each nuclear division is followed by cytokinesis, making it cellular from the beginning. In halobial type, the first mitosis gives rise to two unequal cells. Subsequently, divisions are free nuclear but become cellular after cytokinesis. Endosperm may be completely consumed by the developing embryo before seed maturation, as in many dicot seeds like pea, beans, etc., or it may persist in the mature seeds or may even enlarge considerably, as in cereals, coconut, etc. Development of embryo. The zygote divides into two cells, the upper cell and lower cell. The lower cell divides and forms the suspensor. The suspensor pushes the developing embryo into the endosperm to get food. The embryonal cell divides several times and finally get differentiated into radicle, plumule and cotyledon. The integuments become hardened and thus form the seed coat. Seed coat protects the seed. Thus a seed may be dicotyledonous with two cotyledons or monocotyledonous with one cotyledon. So the seed is defined as a ripened ovule. This is the structure and development of embryo. Importance of seed. It contains embryo which develops into a new plant. The seed coat protects the embryo against dehydration and mechanical damage. Seeds can be stored and transported from one place to another, thus help in dispersal. Structure of gram seed. The seed is enclosed in the pod. It is somewhat conical in shape. The seed is attached to a small stalk. The point of attachment of seed to the stalk is called hilum. Testa is the brown seed coat fused with the inner coat, the tegmen below. It is a small pore, the micropyle. The embryo is enclosed by the two fleshy cotyledons. You can see here monocot. The maize grain is broader in shape. Testa and tegmen are fused together. The embryo is towards the narrower side of endosperm. The endosperm stores starch and protein. The outermost layer, which contains only protein, is called aileron layer. The embryo consists of one large cotyledon called scutellum. The embryo is found inside scutellum. Fruit. A fruit is defined as a ripened ovary. Different parts are edible in different fruits. Significance of fruit: it protects seeds. On decay, fruits which contain chemical substances enrich the soil. It helps in dispersal of seeds. The unripe fruit has a different taste but no smell. But the same fruit, when it ripens, has a good taste and smell. Example: mango, banana. The following changes take place during the ripening of fruit. Starch is converted into sugar. The production of various organic substances gives a different texture, taste and flavor. The breakdown of chlorophyll leads to change in color of the skin of the fruit. Parthenocarpy. When fertilization fails, seeds are not produced, but in certain plants, the ovary develops into fruit like grapes, banana, etc. The phenomenon of development of fruit from unfertilized ovary is called parthenocarpy, and such fruits are called parthenocarpic fruits. Commercial value of parthenocarpic fruits: the fruits are seedless and are hence valued more for eating purposes and contain seeds which cannot develop into a new plant. The fruits contain sufficient growth hormones. Then seed. Seed is the final product of sexual reproduction and seed becomes relatively dry. The metabolic activity of the embryo slows down and in majority of cases, the embryo enter into a phase of inactivity called dormancy or in some cases, if favorable conditions are available, they germinate. Dormancy helps the plant to survive under unfavorable conditions and ensures its germination only under favorable conditions. Germination: embryo lies dormant in the seeds, but when the seed receives the favorable signals and the inputs from the environment, they germinate. Germination is the process by which the embryo grows and establishes itself as a seedling. Step of germination: imbibition of water, and by the seed coat, seed swells up as in get hydrated. Enzyme activity converts the reserved seed coat into soluble form. The seed coat bursts and radicle emerges, and then the plumule grows and develops into shoots. Germination can be of two types: apigeal, where cotyledons come above the ground and form the first leaf of the new plant, like in castor, neem, bean, etc. Plumule emerges from the cotyledon. Hypogeal, where cotyledons remains underground and plumule emerges from the soil and develop into the shoot system like maize, rice, etc. Vegetative reproduction in angiosperms. Reproduction of new plants from the portion of vegetative parts of a plant is very common and is called vegetative reproduction. Stems, roots, leaves, and even buds are variously modified to suit their requirement. This is called natural vegetative reproduction. The new plants formed by vegetative propagation are generally similar to the parents. Natural method. In natural methods, a portion of the plant gets detached from the body of the mother plant and grows into an independent plant. The parts may be stem, root, leaf, or even flower. You have studied about the various modification of root, stem, and leaf in lesson 4 and 5. You have also learnt that these modified portions perform some special functions and also help to overcome unfavorable conditions. The underground modification of stem like rhizome, tubers, bulb and corm are provided with buds which develop into a new plant and are therefore used to carry out vegetative propagation of the plant in the field. Plants with subaerial modification such as pistia, chrysanthemum are also used for vegetative propagation. Similarly, tuberous roots can also be used for propagation as these roots have adventitious buds which ground into a new plant. Sometimes even leaves contribute to propagation of plants. Leaves of Bryophyllum and Kalanchoe have buds on the margin and these buds grow into small plantlets. When detached from the mother plant, they grow into independent plants. In plants like agave and oxalis, multicellular bodies called bulbils develop near the flower. These are called bulbils, which when fall on the ground, grow into new plants. Mode of reproduction, specialized part and example. You can see this in the table. Natural methods and artificial method. Natural method by root, stem, leaves and special parts, and artificial cutting, layering, grafting and tissue culture. So this is the structure with examples. Artificial method. Humans have taken advantage of this natural phenomena and have artificially propagated plants vegetatively by using the specialized parts as described earlier or by cutting, grafting and layering. When we use the vegetative parts of propagating crops or ornamental plants, it is termed as artificial vegetative propagation. Like cutting, many plants like rose, bougainvillea, croton, coleus, money plant, sugarcane, etc. are grown through their stem cutting. Cuttings of these plants can be grown even in water where they strike roots and develop adventitious buds. Layering. In this method, a lower branch of a plant is bent down and covered with moist soil, leaving the growing tip above the soil. A ring of bark is removed from the stem before it is bent down. In a few weeks, time when enough roots have developed on the underground portion above the ring part, it is cut off from the parent plant and grown separately as an independent plant. Example: jasmine, strawberry, grapevine, bougainvillea, etc. Ear layering or goatee is a similar practice where bending of branches is not possible because of the height of plant or due to woody nature of stem. In this method, a ring of bark is removed from a selected branch and it is covered with moist moss and enclosed in polythene sheath. When roots appear, the stem is cut below the roots and planted to form a new plant. Grafting. It is especially important for propagation of seedless varieties of plants. It consists of inserting a small branch into a rooted plant. The rooted plant takes as a stock, is resistant to disease, and is physically sturdy. In this stalk, a branch is inserted, which is known as scion or graft. This scion or graft is the stem cutting from the desired plant. Usually, the grafted end of stalk and scion fit well with each other and are bound firmly with tape or rubber band until their tissues unite and vascular continuity is established. Grafting is mostly practiced in dicot plants. Grafting has been found extremely useful in propagating improved varieties of various flowers and fruits like rose, bougainvillea, citrus, mango, apple, etc. Advantages and disadvantages of vegetative reproduction. Rapid means of reproduction and spread. Offsprings identical to parent. The desired varieties can thus be preserved genetically for use. Food storage organs allow pollination or survival in adverse conditions. Improved varieties of ornamental plants and fruit trees can be multiplied easily. Vegetative propagation is a quicker, easier, and a less expensive method of multiplying plants. Disadvantage: overcrowding and competition for space unless separated artificially. New varieties cannot be produced by this method except by mutation. Disease typical of the species are rapidly transmitted and can decimate a crop. Next and last topic is micropropagation. The technique of plant tissue culture is utilized for propagation of plants. The process is explained below with the help of diagram. A small piece of tissue, organ, or even a single cell is taken from a plant and is transferred to a sterilized container with nutrient medium in aseptic conditions. The tissue grows very fast into an unorganized mass called callus. The callus can be maintained and multiplied for an indefinite period. When small portions of the tissue are transferred to another specialized medium with hormones, it induces differentiation and plantlets are formed. The plantlets can be transplanted into pots and or soil by a gradual process and are grown to mature plants. Advantages of micropropagation: by this method an indefinite number of identical plants can be obtained vegetatively starting from a small amount of parent tissue. In orchids, carnations, chrysanthemum, and asparagus, micropropagation is being successfully tried in some parts of our country. What you have learned in this long PPT, this is the summary. Chlamydomonas reproduce asexually by zoospores and sexually by isogamy. Spirogyra reproduce by fragmentation and scalariform conjugation. In angiosperms, flowers are the organs of reproduction. Temperature and light are two main factors which influence flowering. Stamens and carpels are the male and female organs of reproduction respectively. Male gametes are produced in pollen grains formed inside the anther. Female gamete is produced in the embryo sac in the nucellus of the ovule. The embryo sac contains egg apparatus, antipodal cells and secondary nucleus. Egg cell fuses with one of the male gamete received from pollen grains. Secondary nucleus fuses with the other male gamete. Occurrence of two such fusion is called double fertilization. Pollination is the transference of the pollen grains from anther to stigma. It may be in the same plant or in different plants. Wind, water, insects and animals are agencies of cross-pollination. Wind-pollinated flowers have light pollen grains or bring pollen and the stigma is usually large feather and protruding feathers. Insect-pollinated flowers are usually large, brightly colored, scented and with nectar. Most plants have devices to favor cross-pollination. The zygote develops to produce an embryo. The embryo is present in the ovule, which later becomes seed, and ovary on maturity becomes fruit. Development of fruit without fertilization is called parthenocarpy. Then fruit involves chemical changes in the stored food and pigment of the fruit wall. Vegetative reproduction is the production of new plants from parts other than flower and seeds. Specialized plant parts which bring about vegetative reproduction are as follows: root, stem, leaf, bulbils, etc. Roots by dahlia, stem by underground parts, leaves in bryophyllum, bulbils like in onion. All the above kind of parts have been used by men in agriculture and horticulture as artificial methods of vegetative propagation. Micropropagation by tissue culture enables production of little plants on a large scale. Vegetative reproduction is rapid, easy and cheap. The plants produced are exactly like the parent plant. These all are the practice questions you have to solve yourself in your notebook. Now we will meet in our next session. Thank you very much for joining.